Okay, we might as well start. So happy to see a lot of people in the audience, and was very excited to see so many people sign up to this event, around 80 people all in all. So welcome to Genevia Technologies Spatial Transcriptomics webinar. It's our first webinar and so far has been a success, at least up until this point. So as you might know, Genevia Technologies is a bioinformatics company. We specialize in the analysis of omics data, particularly from new developing platforms such as spatial transcriptomics and advanced analysis like multi-omics analysis, integrating different types of omics data. And this we do for our customers who are researchers in academia and industry, mainly pharma companies. And we employ a team of around 18 senior bioinformaticians who provide this data analysis service, and we've done so since 2011, so we're almost 15 years old now. And I'm Thomas Liuksela. I'll be hosting this event. I've been 10 years now at Genevia Technologies. Started with bioinformatics, now I'm on the business side leading the sales here. And with me here in this webinar, we have two of our senior experts with spatial transcriptomics expertise in particular, namely Doctors Fredrik Salmén and Patricia Thomas. And Fredrik here is a team member of ours for almost 2 years now. He has a long background in method development. He was the original co-developer of the spatial transcriptomics method that was later purchased by Visium or 10x Genomics and marketed now as Visium. He did this during his PhD at KTH in Stockholm. And since he has also developed a single cell method called VasaSeq at the university or Hubrecht Institute at Utrecht. And after that, he worked at a company there called Single Cell Discoveries as an R&D consultant. And I saw there are some people signed up from that company, so shout out to that fine company, Single Cell Discoveries. And now, as I mentioned, Fredrik has been for almost 2 years here in our team, working on single cell and spatial data analysis for our customers in industry and academia. And our other expert, Dr. Patricia Thomas, likewise been a team member of ours for roughly 2 years. When we hired her, she was a PI at the University of Birmingham, managing an independent grant budget of over £600,000 and led research investigating the role of fatty acid metabolism in the development of type 2 diabetes. So that is her academic background. And she's supervised many bioinformatics postgraduate students and lectured on bioinformatics methods at world-leading universities. And in our team, like Fredrik, she's been successfully delivering single-cell and spatial data analysis projects to leading pharma companies and academic institutions worldwide. So, Trish and Fredrik, do you guys have anything to add, any fun facts?
Thank you so much, Thomas, for the introduction. It's so lovely to be here and talking to everybody. I guess a fun fact is that when I'm not doing science, I'm an artist. I have my own studio in my back garden and I've exhibited my work. So that is something about me that is not science related.
Nice.
Nice.
Yeah, thank you, Thomas. Also very happy to be here. It's going to be exciting. Fun facts, when not doing science, I like to spend time with my son, or go surfing or ride motorcycles.
That sounds very fun. So great. We'll dive a little bit, or before diving too deep into the topic, as you might know, the topic of our webinar is successful planning of spatial transcriptomics experiments. But let's just back a little bit and let me ask you guys, what is spatial transcriptomics and how does it differ from normal transcriptomics? What makes it spatial?
Yeah, so traditionally for like bulk transcriptomics or single cell transcriptomics, if you want to study a tissue, you take the whole tissue and you grind it down. For bulk, you extract the RNA and then you can look at like a average expression across the whole tissue, but like sort of in one sample. For single cells, you divide this tissue into like cells or nuclei, depending on, and then you can look at them individually. But what you don't have when doing this, so the sort of what you said traditional way, is you don't have the tissue context. So in spatial transcriptomics, you keep the tissue intact, and therefore you can get the location of each cell and how how the cells are organized in the tissue, how they interact with the neighbors, and so on. Some examples would be like looking at immune cell infiltration in tumor samples, or like neuronal organization, like in brain samples, that are organized in specific layers. All this information is lost when you just mash down the tissue.
Yeah, so just following on from what Fredrik said, there's a really nice analogy within the field, which is a fruit salad analogy, where essentially if you took lots of different types of fruit, so bananas, strawberries, blueberries, and you blended them down, you'd have a fruit smoothie. So you'd be looking at the average expression across all the cells. Whereas in single cell, you can see the individual fruits, you can see the individual blueberries, raspberries, strawberries. But in spatial, it would be more like a fruit tart. Not only can you see the individual fruits, but you can see where they're positioned, what's next to each other. And say I was to add strawberries from 2 different packs, one's really ripe, one's really underdeveloped, we can see those different gradients as well. So we can get a lot more information from that.
Great, I like that analogy. So not only do you learn that you have a cherry and piece of pineapple, but you see how they are located in relation to each other and whether they just happen to be communicating in your fruit tart.
Yes.
So great. So why are we discussing spatial methods here? What will we learn? Would you like to elaborate a little bit on that?
Yeah, I mean, I'm very biased, so I do think that spatial methods are definitely the coolest methods around at the moment, but it's very much an evolving field. Every day we see new packages, new technologies coming through. But the main thing to kind of say is essentially it's a really multidisciplinary workflow, actually one of the most multidisciplinary in molecular biology currently. 50% is the experimental and 50% is the data analysis. And so given the cost of these experiments, it's so vital that when you're planning your spatial experiment, that you start at the experimental and work your way right through to the end of the data analysis.
Yeah, totally agree with Trish there. And hopefully today we will guide you through these different considerations when selecting which technology and how to plan your experiment. And hopefully after this, you have some more insight how to do this.
Great. I do hope I'll have more insight, and I hope the audience will have more insight. Likewise, the bar is set high, but so now a little bit more into the spatial methods. So I know there are different spatial platforms, and within platforms, you have all the different types of parameters and options for your spatial transcriptomic measurements. What are the main types of methods, and what are the differences between them?
Yeah, so
and I believe we should have maybe a visualization of these.
Yes, should be one.
But go on, Fredrik.
Yeah, so there's of course a lot of technologies, different ones, and there's new ones coming out. It's quite a evolving field, but to broadly categorize it, we would divide them into two types. So one would be sequence-based, and the other one would be image-based. The sequencing-based approaches, they are sort of depending on defined locations. They could either be like predefined, like spots on an array, so spatially barcoded spots on an array surface, solid surface. So like Visium HD, both from 10x, or StereoSeq from MGI, or Curio, which is like SlideSeq commercialization. But they can also be like user select regions, such as nanostring cosmics, or the old school laser capture micro dissection, where you cut out a region with laser and collect it.
Yeah, you probably meant geomics, not cosmics.
Yes, of course. Sorry, geomics. Yes, cosmics is an image-based one that Trish will tell you about a bit later.
Nick Meister.
So these methods, what they are, they're typically quite broad. They provide like whole transcriptome information. So, good coverage of genes. They have this at either spot level or selected region level. And because they are so broad, in terms of gene coverage, they're very suitable for like exploratory analysis, discovery-driven studies, where you want to see like large-scale patterns across a tissue.
So yeah, just to follow up on that, if we can see to the right side of visualization, we're looking at imaging-based methods. So essentially what you're doing within imaging-based methods is you're detecting RNA directly inside the tissue using microscopy. And the benefits of really doing this is that you get to maintain the tissue structure and the spatial context. So overall, what we do is you essentially add your probes, amplify your signal, take multiple images, and then the software of the sequencer would mostly decode the barcodes. And at that point, you'd pass over the data to a bioinformatician who could then analyze it. So some of the advantages of this is it's very, very high spatial resolution. So we're looking really more at the single cell level than the whole tissue level. And so some of these new technologies are not only looking at the single cell level, they're also looking at subcellular. So to kind of give you an image in your mind of what that looks like in terms of data, with bulk or single cell, you have a counts matrix, which is essentially your genes as rows, your samples or cells as columns. But in spatial data, you'd have your X and your Y coordinates of where that cell is located. And using the subcellular, you'd also have the Z coordinates. So for any microscopers out there in the audience, this is quite similar to your Z stack. So we'll be taking that information. And it's really best applied to very, very specific pertinent biological questions. So things like cell-cell interactions, tissue organization, spatial gradients, local microenvironments, that would all be able to kind of look at using these imaging-based methods. Some of the examples that you can have is things like xenium, cosmics, which Fredrik already mentioned, burfish. But we will say that although you can fit them into these very broad categories, there is obviously crossovers as these technologies evolve. So for example, in cosmics, they have now brought out a whole transcriptome for human tissue. So yeah, there are crossovers within that.
Great, thank you. So for me, it sounds like in a very coarse-grained scale, the sequencing based methods are sort of like out of, if you think of bulk RNA sequencing, but added the spatial coordinates. And with the imaging based, it's kind of born out of microscopy, where in addition to just the images, you get the transcripts identified within cells.
Yeah.
High resolution. And you mentioned resolution as one of the features of these methods that you do want to take into account when picking the best method for you. What are the other features besides resolution that matter with these methods?
I mean, so with all experiments, it very much depends on your biological question, obviously. But some of the things that you want to be thinking about are how much spatial detail do you require? What number of genes are you wanting to measure? Also the type and the quality of the tissues. When I was working in lab and I did sectioning, sometimes I'd transfer that tissue to a slide, it would fall apart. And obviously in spatial, you're going to be losing whole chunks of data doing that. So you need to be kind of thinking about things that are quite logical, really. And also, can your biological question be answered at the level of the tissue region, or are you wanting precise localization of specific cells and their interactions? And thinking about all these things together will kind of guide whether you're wanting to use a sequencing-based method or an imaging-based method, and which one essentially is most appropriate.
Yeah, how about Fredrik? What are your favorite parameters when picking your platform?
Yeah, first I want to say I recognize what Trish said about the tissue falling apart. You know, when you try to make the perfect experiment, never really happens, right? But another very important aspect, I think, is the cost of the technology you're going to use. So most of these technologies are quite expensive. So you have to take that into account. You know, some of them are less than others, and you have to take your budget into account there. But also availability. So, you know, which method is available at your department or institute, or, you know, if you have an instrument in your group, perfect, you know, then you can run whatever you want. Most don't have that, right? So you have to take into consider the cost and if you can actually, you know, run it within a reasonable time frame. So just to take an example here, so let's say now that you are one of these groups that doesn't have an issue with the money, and you need a whole transcriptome, and you need the highest resolution possible, you know, of course, then you can go with like NanoString Cosmic's whole transcriptome platform, or Visium HD, or StereoSeq, you know, there's sequencing based with like very, very good resolution. But if you don't have that kind of money, you want to start like a smaller pilot, maybe try something like Curio, for example, you know, SlideSeq, where you can run only a few samples, so that brings the cost down. But if you need like full single cell resolution, but still on a budget, then you can look at some of the image-based approaches like Cosmics, Xenium, or Murfish, but using a smaller panel. So not going, you know, with the top panel, maybe like 500 or 1,000 genes or so. Yeah, that's basically my input on that.
Okay, so it adds a little trade-off, the cost. So in addition to spatial data, you might be left with some extra space in your wallet, depending on how expensive methods you want to use. At this point, I want to remind you something I forgot to say in the beginning. There's a chat box, and you can add questions there to Fredrik and Trish about spatial transcriptomics or anything else. And at the end of the webinar, we'll have time to address a couple questions. So feel free to ask questions there. And then back to questioning our experts. So we talked about parameters such as the resolution and cost. Something you mentioned, Trish, was the amount of genes. So what does this mean? I understand different platforms and different types of panels within platforms come with different amounts of genes or transcripts to target. Now, what does that mean for me when I'm studying my favorite tissue type? How do I know what resolution to use and how many genes do I need to target? Is it okay to go with this 2 genes, or do I need a whole transcriptome wide approach?
Yeah, so 2 genes, I wouldn't say no, but you know, wouldn't be too positive either there. But I would say if you have a clear view of the tissue type, you know, it's, you know, the question that you're going to study, it's like a very studied tissue or disease type, then it's easier to pick a panel from the ones that exist, and then maybe add some gene on top of that. So most of them also allow that, you know, you add some custom genes to it. Now, if it's more like an exploratory experiment, you know, very novel tissue, novel disease, or understudied, it's going to be difficult to pick like one of the smaller panels because you don't exactly know what to expect there. So then you might have to go for a larger panel, and it's going to, you know, add cost there. But yeah, specific biological questions, well-characterized tissue, smaller panel, it's totally fine as long as you are, you know the genes are there that you're interested in.
Yeah, so to give an example from my academic days, I used to work in pancreatic islets, so islets of Langerhans, and they're predominantly made up of 5 major hormone-secreting cell types. So if we use this as examples, I've taken this from a paper, Howell et al., and this is the top panel where they've used a sequencing-based approach. These guys essentially were doing method development. They were finding, I think, that insulin was really overpowering signal. But what they wanted to do essentially was just highlight the main canonical cell types within there. So they just used things like insulin, glucagon, somatostatin markers, and to see where the individual cells were. Whereas this example I'm giving below, which is a visual-based one, this is from Torres Cano, and they used a custom panel, an image-based method, because they were looking specifically at mesenchymal lineage in pancreatic development. So as you can see, again, it very much depends on your biological question. Are you wanting to really look at, the nuances within that data, or are you just wanting a broader view?
Okay, great. So, well, let's imagine I want to use a transcript panel. I don't need to go transcriptome wide. How do I choose the right panel? I imagine different platform providers have different types of panels and customization options to these.
Yeah, so I mean, this is such a growing field at the moment, is choosing the right panel and how to do that. So essentially, the obvious one is that you match your panel to your biological question. So if I'm studying cancer, I would use a cancer panel. So it's quite goes without saying. And as Fredrik said before, you can add things in, so you can kind of grow that panel a little bit to include genes with both cell identity and biological states. So progenitors, if you were looking at, say, TNF alpha pathway activation, you would include genes for that. So there's obviously the biological aspects of it, but you're also wanting to account for technical constraints. So at the moment, and this won't always be the case, but presently most platforms can only measure a certain number of genes. And probe design, another growing field, it means that not every gene can be targeted as well. So some genes, you can't really make the probes for them because they cross-hybridize with other genes. Also, I mean, if we look at our previous example, so if you're looking at the islets of Langerhans, well, 50% of those islets are going to be beta cells secreting insulin. So that's going to really dominate your signal. And so if we wanted to look at something like PDX1 alongside, which is a transcription factor, it's going to be a lot harder to see that. So essentially, just be aware that your very highly expressed genes can really dominate that signal and reduce the sensitivity of others. When you're designing a panel, also you want to think about designing it strategically. So avoid genes that can't really be reliably measured and really prioritize very informative markers, so over more redundant ones. And also, when you go to a manufacturer who's making these panels, remember, you know your biology so much better than they do. You know your biological question. And so don't underestimate the power of your own knowledge as well, and use that knowledge and use your hypothesis to kind of build that. We can also do things like using single cell, things that have come out of the single cell world, and bioinformatic tools that can essentially help you refine your panels. And yeah, so there would be some of the things I'd think about when I was doing that.
Great. Fredrik, do you have anything to add?
I'm going to be boring, sorry, again, and mention the cost,
Very important.
Yeah, very important. Unfortunately, you have to think about how much you're going to spend on the experiment, because there are going to be more costs to it. I mean, if money is not an issue, yes, go with full panels, but to take an example here without mentioning exact numbers, because that's going to vary depending on which part of the world you're in, which institute and so on. But if you look at NanoString, CosMx panel, taking the whole transcriptomics panel, that's 4 times more money per section than running the really small one, not really small one, but the smaller one, 1,000 genes. So you can think there, does the smaller panels contain enough information there? Then you could actually run 4 times more samples for the same money.
Okay, so a real trade-off there. Do I need all the genes, or do I want more samples to get more power for more statistical power for my analysis?
Exactly.
Well, talking about analysis, we are bioinformaticians after all, aren't we? Do these methods come with built-in analysis solutions? Now, if I start using spatial transcriptomics, I generate some data. What do I need to understand about the bioinformatics? Do I need to have a bioinformatician, or can I just press the button and get results out of the sequencing machine or spatial platform?
Yeah, so I could answer probably yes to both your questions, but that would be, contradictory. But I would say, coding-based approaches and this type of online platforms, they both have strengths and limitations. And what you choose depends a lot on, the complexity of your data, the need for flexibility. Do you have a lot of samples? Do they need to be integrated, for example? Is it a very standard clustering, or do you actually need to treat the data in other ways? It also counts in your coding experience, So online platforms, they're more easy to use. It don't really require you to know coding, although some of them you can actually upload your own scripts to them. Usually they have like built-in visualizations, reporting, which makes it pretty good for exploratory analysis. Quickly you can look at the data that you generated. But overall, I would say that they're more suitable for like an experimentalist that want to take a quick look at the data themselves, which is of course very exciting. You want to see the whole process of your data
As fast as possible.
As fast as possible, yes. You want everything, of course, right? The problem though is they are less transparent usually and reproducible. They're a little bit of a black box. You don't know exactly what's going on under the hood unless you upload your own scripts to them, but then you need the coding anyway. And they're also not really suited, to output that, that perfect figure or table that you're going to need for your publication in the end.
Great. Trish, do you agree or disagree?
Yeah, 100% agree. So obviously, coding-based approaches, they are much more flexible, they're more transparent. Reproducibility in science is such a huge thing. So they are rightly so, and so they're also reproducible. And so it makes them really well suited for very large and complex data sets that really need a custom analysis, of which spatial data inherently is. It's inherently large, it's inherently complex, it's very noisy. So we do need that for custom analysis. Obviously, as Fredrik said, the main trade-off is that you need to be able to code, and that is a very steep learning curve when you're first learning it. I think it would be naive not to kind of bring in AI here. Obviously, we have very powerful AI tools that are coming through, and they make coding much more accessible, but they don't really replace understanding. So, for example, you still need to be able to interpret that code. You still need to understand the underlying statistical principles and really collaborate with bioinformatician. You are still going to need to write this up, publish it in a paper, and a statistician will still go through often your paper to review the methods. And so AI doesn't always get that right. And so it's quite a risky approach to be taking. So kind of coming up with a slightly more concrete kind of example between the two. Recently, I was working with the most phenomenal PI. She was amazing. And she was looking at cosmics data in prostate cancer. And so she had used a graphical user interface from one of their top providers. And when she came to me, she'd identified 8 different cell types within her data. But by using coding-based approach, I was able to identify 15 different cell types, including new cell types that we didn't even know existed in prostate cancer. So that's really the power of the coding method. So yeah, just to back up really what Fredrik was saying, if you're wanting that quick and dirty look at your data, what does your data look like? They are absolutely brilliant. But if you're wanting high impact visualizations, analysis, you do really need a coding approach.
Well, I'm beyond relieved to hear that senior scientists with computational skills are still needed. Now let's imagine you, Trish or Frederick, you get a new batch of spatial data. As a data analysis, what do you have to look at? Are there like quality issues or any technical things that you need to absolutely take care of or look at? Are there some like considerations, quality issues, or other with spatial data in general?
Yeah, so I think because this is a new and developing field, there really isn't a gold standard workflow. We're still essentially working it out. But there are some core checks that I do when I got the data. So I really need to start off by assessing whether that data is technologically sound and biologically meaningful. And so what most people, I think, don't really realise, actually, in these types of analysis, about 60 to 70% of what we do is actually just cleaning up the data and making sure that we actually get a biological signal that we can trust from it. So very, very first thing I do is, so if I have just generated a spatial data set, I wouldn't try and store that on my laptop, because my laptop would not survive. It would use up so much memory. So we use something called a high performance computer. And so this essentially is just a cluster of computers. So you've got much more memory there. And so what we do is you would send us your data. And so when we got the data, actually just that transfer process can corrupt the data. So I would make sure that all my data had transferred correctly, it wasn't corrupt in any way, and that everything was present and in the right place. And then I'd move on to things like my quality control checks. So this would include things like your genes and your counts per cell. So sometimes the technology might not have read a particular cell very well. That's not really going to be useful to me, and it'll just add a load of background noise I don't really need. I'd also look at things like mitochondrial gene counts. So if you have a very high expression of mitochondrial genes, it's indicating that essentially those cells are dead and dying. So we don't really want those in our analysis either. We'd also check things like background noise. So we'd use the negative probes for this to make sure that we're not just seeing a load of noise. And we'd also check things like segmentation. So some of the new packages coming through, like Space Ranger version, I think 4, is doing the cell segmentation for you. So what I mean by that is the algorithm is actually identifying the outline of the cell. It doesn't always get it right. And so sometimes you can have 2 different cell types in there. So you need to make sure that your segmentation is correct. Beyond computational, I would also try and work really closely with the experimentalist. There are things that the experimentalist can remove slightly easier than we can. So things like batch effects and putting different patients on the same slide, things like that. And then one final kind of point I think about is if I was using a new technology. So a few months ago, I was working with a PI here in the UK, and he had gone and used Visium HD, which is so new in its life cycle. And so the first thing I did actually was to reach out to the manufacturer, talk to the manufacturer, say, is there any quirks or biases in this data that I should be aware of before I go in? So I'm so used to looking at quality control checks, but quality control checks might be very different for a new technology coming through. And so also looking at published data sets, see what their quality controls look like. So what might be normal for one technology might be very different for another technology. So I'd be thinking about those kind of things within quality control.
Well, there's definitely a lot to think about.
A lot to think about. It's a very big part of this analysis. Yeah.
And obviously all different platforms have different issues. So like with imaging-based methods, the segmentation is a question.
Yeah.
And so on. Well, moving on more downstream of the analysis, I have one quick question specifically about, someone mentioned already, using single cell data in the analysis of spatial data. So let's say I have single cell data from my tissue type of interest, and I'm now doing spatial experiments. How do I use that single cell data, or can I use that single cell data in the analysis of the spatial data?
Yeah, so I mean, single cell, for example, it's a huge field. It's come on a long way. We have a lot of data there, much more than we do of spatial. So we can use it for things like probe design and panel selection. One of the main things I actually use it for, though, is essentially we have a lot more of something called zeros and dropouts in the spatial data, so gaps in the data. So this becomes a particular problem when you're trying to annotate the cells that you're looking at. So essentially what we can do is we can use machine learning methods to transfer cell type annotations from single cell data and project them onto your spatial data set. So super important.
I agree there. Yeah, probe design, very important if you go for image-based technologies. And like Trish said, there's a lot more single cell data out there. The atlases, you can use them for cell typing. So together with spatial data, it's a very strong combination because you have a lot of information there, with good molecular resolution, so more counts, more genes per cell. And then with the spatial, of course, you have the spatial context. So you get more meaningful information from that.
Okay, so it does add value. Well, just a quick follow-up question to that. How about with spatial data? Now, if I'm doing spatial experiments, I'm paying way more for those experiments compared to single cell experiments. What value does the spatial part add? So essentially, what are the analyses that answer research questions that single cell data alone can't answer?
Yeah, so once you've done your QC, you've normalized, you've annotated your cell type, essentially that is where the fun begins. And there is a lot of very cool methods coming through. So because you have every cell or spot, if you're doing kind of sequencing ones, it's embedded in physical space. So you've got things like distance, neighborhood, tissue structure suddenly becomes such a big part of your analysis. And so with that information, you can identify tissue niches, microenvironments, which cell types co-localize with which segregate, is there boundaries there? Are they sharp or graded boundaries? So there's so much you can do with that.
Yeah, I agree with Trish there. The spatial data enables true cell-to-cell communication. For example, you can look at the receptor and a ligand. Where are they expressed? Is they far away? Are they close to each other? And like Trish mentioned, are these genes expressed as patchy hotspot regions, or is it more like a gradient pattern across the tissue? So yeah, that's very, very important thing that you can do with spatial. And one final thing, I think that's also important to mention it. In single cell data, sometimes you get sets of cells that look very similar, on a molecular level. But once you look at them in the spatial context, you see that they actually take up different parts of the tissue.
Yeah, so if I have in a given cell type, I have a continuum or different subclusters with single cell data alone, I might not know if those are mixed in the tissue or if the different types of cells are just in a different part of the tissue.
Yeah, exactly. It might look like a blob, in the UMAP of the single cells, and you can sort of subcluster it, but you're not convinced. But then you put them back on the tissue and it's completely different regions.
Okay, well, great. Thanks for that. Now, for the latter part of the webinar, we wish to learn a little bit more about our audience members' experience with spatial methods. So we'll have a short questionnaire, takes maybe 1 minute max to fill that out. And you'll see a link to that questionnaire soon. And I ask you to please fill it out. It's important for us to learn better what sort of backgrounds you guys have with spatial methods, if you're still planning spatial methods or use them for a long time. We'll turn our cameras off for a minute just to let you answer that questionnaire, and don't go anywhere. The best parts are still ahead of us. Okay, we're back and live and alive. Great. So as promised, the best parts are still ahead of us, the real dessert, the pie tart, as I call it. So a few good questions still. So we've talked about the different types of platforms, different parameters, different features of these different platforms to consider when planning an experiment. Considerations from the data analysis part to take it to think about when you are planning your experiments. Well, how about, it's become clear by now that this is a very rapidly developing field. New spatial methods are being developed all the time. So what do you know about that? Where is the field developing? What other spatial approaches are being developed?
Yeah, so it's super fast moving. Like I said earlier, if we had this webinar this time next year, it would look very different to what it does today. But some of the things that you're thinking about, the developments essentially along some major axes. So spatial multiomics is a really big one. So moving beyond RNA-only measurements, and we're wanting to capture different molecular layers. So things like proteins, chromatin accessibility, epigenetic marks, metabolites, for example, but still whilst preserving their spatial context. And so one of the issues that, well, not really an issue, I think we're kind of moving towards is that currently when we analyze the data, we're looking really at proteins and RNA or whatever else you're looking at than the RNA in parallel. But what we're wanting to move towards is really fully integrating them. So jointly modeling how transcriptional programs and, for example, protein levels interact.
Okay, so more omics layers and more multi-omics. Well, how about Fredrik?
More omics layers, more integration. Yeah.
More
Where
of
does
anything,
Fredrik see the future?
Yeah, so I mean, all these new technologies, they also open up the door, to like spatial readouts directly in the tissue. So now you can look at, effects of, for example, like CRISPR edits or drug treatments or environmental stimuli. So, it shifts the field from more like purely descriptive into like in situ experimentation for real at this point, Then, of course, all technologies always have to improve on everything, so scale, resolution, the image base, they're already down at subcellular. Like Trish mentioned, you have X, Y, and even Z coordinates, can be very useful in a lot of questions. They are improving the segmentation constantly. It's still an issue, it's sort of like creating some sort of contamination. So we improved segmentation. It's very, very helpful. Gene panels, also like Trish mentioned and I mentioned before, they're becoming bigger, covering the whole transcriptome even, moving to more organisms than just human. Then, of course, something that's going to be very important, I think, it's to be able to have more high throughput. So in terms of larger tissues or more samples per run, because, at this point, it's quite time consuming to run and costly. And, you want to create a lot of data quickly so you can analyze it. So I would say, yeah, it's a balance between targeted and unbiased approaches. The targeted, they're becoming more flexible, better optimized. The whole transcriptome ones, they're more like improving the sensitivity and throughput and cost efficiency.
Okay, so the technical parameters of the existing platforms are being developed, more resolution, sensitivity, larger panels, more omics data types, more application areas for these measurements. Yeah, and I think like especially for testing like drug effects and mechanisms, when you do it in an actual tissue and you see how different cell types are affected, you might save a lot of time and money when you do it that way rather than testing an individual cell types alone. And of course, it's a way more realistic case when you see the actual tissue and actual organ, how the different cell types there interact or change their interaction in response to a drug. So, okay, so that's how the platforms are developing. Well, how about the bioinformatics side? Is there any development going on in the computational part of this all?
A huge amount. So one of the things that we need to do in spatial is very much keep up to date with the literature, as new tools and methods are coming out almost daily. It is very fast moving. So I code predominantly in Python, and so the Python community have developed, specifically Cyverse, some amazing machine learning algorithms. So these are really keeping pace with experimental technologies. And so some of their applications, so it's for data integration, pattern discovery, tissue organization, and improving image processing, essentially. Also, these data sets, they're getting larger. You know, I said before about keeping on your local machine, you know, the memories that they require is huge. So when you're analyzing them, the computational time is also, the bigger the data set, the longer the computational time. So they're getting a lot better at handling these really, really big data sets with increasing spatial resolution. And I think one of the things that, another area that would be spatial atlases. So currently in the single cell world, they've really built these huge atlases. So instead of just doing one lab doing one data collection, another lab doing another, is that they integrate them all together. And so this is much more powerful, right? You have much more information, you have much more data, and so you can get much more cell types, etc., out there. And so we're wanting to do the same with spatial. We're wanting to make these spatial atlases. So overall, spatial it's becoming broader, it's becoming deeper, it's more integrated, and yeah, it is such an exciting time to work in this field. I really can't stress that enough.
So more advanced methods, but also better data resources for like annotating your cell types and spots. Well, just to wrap up this part before moving on to the questions, could you very briefly just summarize what do you think are the most important factors or the most important components of a successful transcriptomics study, if you could name just a couple of them?
Yeah, so I would say in a perfect world, which this never is, but we should strive for that still, you know, try to plan the entire workflow. So like from the experimental design all the way to how the data is going to be analyzed and who's going to analyze it, you know. So because the type of question you have will also influence the technology that you choose, as we have, you know, talked about today. You know, if you have a well-defined biological question, you know, with the study design, technology selection, and downstream data analysis, it's going to be much smoother, the whole process. And you're going to really avoid this, you know, this type of thing that you generate data, you know, you just run something, you generate data, and then in the end, you spend time and money, and it turns out this was the wrong type of data for your question, you know. That's a very painful lesson to learn, but unfortunately it happens, of course. From an experimental perspective, I will also think about, you know, have appropriate controls for your study. So the negative and the positive, depending on what you're looking at. Sufficient biological samples, of course, getting more statistical power. So it's not just a case study. And then something that is very, very important that I think a lot of people sometimes forget is the quality of the specimen that you're using. That is very, very critical. Because if you have a sample, it doesn't matter if it's very precious and very hard to get. If the RNA is degraded in that sample, you know, there's not the best technology or the best data analysis in the world. You know, you can't save that. You can't magically get the RNA back. That is
even you can't save it, Fredrik.
You know, I can't save it. No, if it's gone, it's gone. So if you can, I would strongly suggest that you really validate your sample before you run it through a whole process, you know, a whole workflow.
Great. How about Trish?
So I don't really have much more to add to that, but one of the key things that I will add is communication. Communication. It's so key. You have to have a shared language between the experimentalist and the bioinformatician. And so if you can talk on each other's levels, what you can do is streamline experimental data. You can come up with a feasible statistical analysis. you can make sure that you've chosen the right technology before running it, and that the data type is really appropriate for the biological question. And so this gives actually much more realistic expectations around data interpretability. I'm losing my words. So if you include a bioinformatician, so from really early on, it can also do things like we've mentioned money. And so if you're actually applying for a grant, if you speak to the bioinformatician first, there may be hidden costs there that you haven't necessarily taken into account. So things like your computational analysis, is that going to cost? Do you need to pay for a bioinformatician to do it? And even things like data management costs. So, you know, where are you going to store this huge data, and is that going to cost you, and how many terabytes do you need to buy, and things like that. So also, finally, something to think about is you can't, if you're trying to publish spatial data, you can't just rely on the computational findings. Those computational findings need to be validated as well. So how are you going to validate what we found within the analysis to ensure that what you're presenting in a paper is meaningful and robust? So there are lots of things to think about.
Great. I love that you mentioned communication. Spatial transcriptomics is not just about cell communication, but it's also about scientist communication. And I guess we are in the business of convincing people that even bioinformaticians can be great at communicating.
Absolutely.
100%.
Even you, Fredrik.
Even me. Even me.
Great. So now we still have a little bit of time for some questions. I see we have a panel of questions. We'll try to tackle a couple of them. There's a fast and easy one first. If I have to know coding for analysis, which language would you recommend?
Ooh, I think that's a perfect question to be asking, a very loaded one. So I code in Python. Fredrik codes in R predominantly, so we often have the debate of which one's better. I think, to be honest with you, if you're analyzing in Python, it's a lot more user input. So our packages are generally much more tailored for people who are just starting out in the field. But also our visualizations are way better than Python visualizations. So I think if you're wanting nice, pretty figures, actually doing both Python and R is better. And Python, as I said before, is really good for machine learning, and it's really come on in that field. So it really depends on what you're doing and what you want out of it. And yeah.
I agree with Trish. I think also that Python is a more logical language than R. So that's something to consider. You know, it handles big data much better. You can get crazy sometimes with R, you know, when it's just crashing. But yeah, like Trish said, visualization better. So ideally you run both, you know, you run the big data sets in Python, and then you can show the visualizations in R, for example.
I'd also say, sorry, just to add on to that the algorithms are often different in Python and R. So it very much also depends on your data and what you're doing. So yeah.
Definitely, yeah.
Okay, so I guess the verdict is Python or R.
Yes.
But on a cluster, not on your local computer.
Not on your local computer.
Please.
Well, I have a specific question from the audience. How often do you use third-party segmentation tools? If you have used third-party segmentation tool, how easy it is to import, for example, into Xenium Ranger? Have you guys ever done it? Use some standalone segmentation package?
I haven't. Most of the ones that I've come across have been in some sort of user interface, and obviously I'm obviously going to code because it's a lot quicker than that. I will say though that you kind of make it work. Whatever data type that you get, you furiously head to the literature, start reading up about it, talk to your sysadmin, and yeah.
What if I don't have a sysadmin?
Position, and they can help you.
Then I guess you have to contract a bioinformatics company that has a sysadmin and on call.
So I haven't done it either. Tried a lot of tools, but I would say it is that, without saying too much now, but I think especially if you work with an image-based technology, you know, you shouldn't think about only the cell staining as the segmentation. Think about that you have X, Y, and Z coordinate of transcripts as well. that can be used to improve segmentation and correct things. So think about all the information you have there in your data, not only what's the segmentation, things that the algorithm thinks that the cell is, or which are two cells, which are one cell. So.
And just to jump on that as well, I mean, like we've said, spatial methods have been developed, but we can also take things from other fields. So, for example, geographical packages, there's a wealth of those, and rather than dealing in meters, centimeters, we can deal in pixels if you're doing image based. So don't be afraid to look at other fields and what they're doing as well, because you can often kind of take some of those packages and use them.
So tissue is a literal landscape.
Yes, it is. But make sure you do.
Rivers, mountains and stuff. Cool.
Yeah.
I have still one question. How do you detect things like spatial gradient of gene expression? And the person says, I have done a bit of single cell RNA-seq analysis, but spatial. So I guess from single cell, we know these like trajectory analysis of gradients from one cell type or state to another. How about with spatial? How does a spatial dimension add to this?
Fredrik, do you want me to first? Or?
Yeah, you can go.
Yeah, so essentially, this is quite a new field or a new type of analysis. So without trying to get too technical, essentially you can make a neighborhood graph. So you can put all of your cells into clusters and groups, and then you can say, okay, well, what does my transcriptional expression look like inside that cluster, and how does that compare not only to the clusters but also to boundaries? So these boundary cells, do you have bleed through coming through, and are these genes unique? And so by doing that, you can then look at these gradients and look at how the tissue, you can actually just visualize how the tissue looks just according to their transcriptional expression.
Yeah, I agree. I mean, what the important thing here is that you have the spatial physical distance there, right?
Yeah.
So you can use that in this gradient, measure like how strong is the signal on, XYZ distance from something.
Yeah, and how
in single cell data, you only have the molecular distance, right? Doesn't show where it is in the tissue.
Yeah, so in single cell, within a cell type, you could see there's a gradient of whatever cell type or cell state. But with the spatial data, you could see that, well, actually the cell state gradually changes from, say, in cancer, like from the tumor center to the outer parts of the tumor.
Yeah. By physical distance,
This is one of the most powerful things about spatial as well, because if you're like Fredrik said, if you're just looking at single cell, you can say, okay, well, this pathway changes, but suddenly when you're visualizing that on a tissue and you realize how heterogeneous even just one cell type is, I mean, it's beautiful and amazing.
It is beautiful, and all beautiful things must come to an end. We are almost at the end of our webinar now, and I don't have any more questions. We didn't have time to answer all the audience questions, and I feel your pain. I have a lot of questions that I didn't have time to ask Fredrik and Trish, but we did have the questionnaire in the middle of the webinar, and we will be in touch if you leave your contact details there, and if you want us to schedule a meeting with us to talk about your experiments, if we can help in planning your experiments or data analysis, or even helping you and analyze your data, we'll be very happy to talk in more detail. This was a very, very limited preview of what our people and our expertise is about. And finally, I just want to thank everyone, our audience. Very happy so many people showed up, and our experts, Trish and Fredrik, and my colleague Johanna, who organized this all behind the scenes. And do follow us on LinkedIn if you want to know about future webinars and other events. So thanks a lot, and hopefully see you guys in another context in maybe follow-up meetings.
Thank you. Bye.
Thank you. Bye.